Madurella mycetomatis is a fungus primarily reported in Central Africa as a cause of mycetoma in humans. It has been misclassified for many years, but with improvement of molecular techniques, its phylogenetic classification has been established. Many methods exist to identify mycetomatis, both in lesions and in culture. Histological examination is especially useful, as it has many unique morphological features. Strain level differences in response to antifungal agents is informative for treatment and laboratory isolation of cultures. History. Madurella mycetomatis underwent many name changes. In 1901, Brumpt described the first recorded case of mycosis caused by mycetomatis, identifying black granules in association with mycetoma. In 1902, Laveran named the fungus Streptothrix mycetomi, which he had identified from a mycetoma grain. In 1905, Brumpt corrected its genus to Madurella, in turn changing its name to Madurella mycetomi. The fungus was grown in vitro by Bro in 1912, so it could be studied in culture. In 1977, the British Medical Research Council changed the name to the currently accepted name, Madurella mycetomatis, with the binomial name determined. Mycetomatis still remained incorrectly classified in the Pleiosporales. This error was eventually corrected, and mycetomatis was placed in the Sordarieles order. Phylogeny. The genus Madurella contains only two well-defined species, mycetomatis and grisei. Roughly a dozen other species of uncertain validity have been described as genus Madurella based on in vivo similarities and cultural sterility. Although similar, there were important physiological and morphological differences between the two well-defined species, leading scientists to doubt their phylogeny. The development of ribosomal sequencing and other molecular techniques led to the discovery that mycetomatis did not share a common ancestor with grisei and that mycetomatis belonged in the Sordarieles order, further confirmed with comparative genomics of Chaetomium thermophilum, a known species of the Sordarieles order. Genotypic variation can help explain geographical distribution of fungi and differences in host symptoms. Restriction endonuclease assay, RAIA, and random amplification of polymorphic DNA, RAPD, helped characterize the different genotypes of mycetomatis, namely 2 genotypic clusters in Africa, and 7 different genotypes from other continents. Further testing with amplified fragment length polymorphism, AFLP, found 3 clusters in Sudan, in contrast to the 2 clusters identified by RAPD, proving AFLP to be a more sensitive method. This showed that mycetomatis is not genetically homogeneous, and explained the variability in host symptoms affected by the same etiological agent. Physiology and ecology. Madurella mycetomatis has been identified in both soil and anthill samples, growing optimally at 37 degrees Celsius. However, can viably grow it up to 40 degrees Celsius. This ability to grow at high temperatures is a feature that can be useful in identifying the fungus in culture. The fungus's ability and inability to break down various molecules can also be used to confirm its identity. Madurella mycetomatis is amylolytic yet is only weakly proteolytic, and has the ability to assimilate glucose, galactose, lactose and maltose, while unable to assimilate sucrose, potassium nitrate, ammonium sulfate, asparagine and urea can also be used by the fungus. Madurella mycetomatis produces 18-dihydroxynaphthalene, a precursor to melanin, a protein extracellularly attached to proteins. Both molecules are responsible for the characteristic dark grain color. The melanin produced by the fungus has also been identified as a defense mechanism against processes such as hydrolytic enzymes, free radicals, redox buffering, antibodies and complement. The fungus also produces pyomelanin, a brown diffusible pigment. Growth and morphology. The growth of M. mycetomatis is very slow and can be broken down into 3 stages. Initially the colony is dome-shaped white-yellow or olivaceous brown in color. The mycelium is covered in gray down, giving it a woolly texture. Following the initial stage, brownish aerial mycelia, 1 to 5 micrometers, form and the colony starts producing a diffusible pigment called pyomelanin, and becomes smooth in texture. Older colonies form masses of hyphae called sclerotia or grains. In nutritionally deficient of potato-carrot media, black grains, 0.75 to 1 mm in diameter, with undifferentiated polygonal cells can be observed. Grains of M. mycetomatis are hard and brittle, ranging between 0.5 and 1 mm, maximum being 2 mm, with masses from 2 to 4 mm. The grains are oval and often multi-lobed. They are reddish-brown to black in color and texturally smooth or ridged. The grains are made up of an internal mass of hyphae, 2 to 5 micrometers in diameter, with terminal cells swelling from 12 to 15 micrometers, maximum being 30 micrometers, in diameter. Overall 2 main types of grains are observed. The most common type is compact or filamentous, where a dark brown cement-like amorphous, electron-rich substance fills the voids surrounding the hyphal network. The hyphal network differs in growth between the cortical and medulla region, with radial versus multidirectional growth respectively. When stained with hematocylin and eosin it appears rust-brown in color. In contrast, the second type, vesicular, has a light-colored medulla and a brown cortical region filled with hyphae and vesicles 6 to 14 micrometers in diameter. Often it is difficult to determine the transition point from cortex to medulla. Lesions can have both the filamentous and vesicular type grains at the same time, although conidation, a form of asexual reproduction, in M. mycetomatis is rare. 2 main types can be described in vitro. In the first type, oval to piriform conidia, 3 to 5 micrometers can be observed. The conidia have truncated bases and are on the tips of simple or branched conidiophores. In vitro, this type of conidation can be observed in 50% of cultures on soil extract, hay infusion or water agar. When grown on potato carrot agar or cornmeal agar, the second type of conidation is observed. This type is characterized by small spherical conidia, 3 micrometers in diameter, on tapered tips of flask-shaped phyalides and collarets. On SDA media, M. mycetomatis is sterile. No sexual stage has been identified for M. mycetomatis. Mycosis. Madurella mycetomatis is the most common fungus with respect to causing mycetoma in humans. A chronic localized inflammatory disease. Madurella mycetomatis accounts for 70% of mycetoma cases in Central Africa, especially common in Sudan. Cases of mycetoma caused by this fungus have also been reported in West Africa, India, Venezuela, Curaçao, Brazil, Peru and Argentina. The mode of entry for etiological agents of mycetoma, such as M. mycetomatis, is trauma, such as snake bites, knives, splinters, thorns and insect bites. Thus, having identified M. mycetomatis in soil and anthill samples substantiates its involvement in mycetoma. Infected hosts from samples in Sudan show variability in clinical symptoms. This corroborates the heterogeneity of M. mycetomatis genotypes. Detection. There are various methods available for the purpose of differentiating fungal species. Histological examination allows for the exploitation of unique morphological features of M. mycetomatis, including but not limited to the unique cement-like substance and multi-lobed morphology of the grinds. Molecular analysis allows for a more sensitive technique to discriminate between morphologically similar species. An internal transcribed spacer. Is amplified by polymerase chain reaction, PCR, followed by restriction fragment length polymorphism, RFLP, digestion or gene sequencing to obtain the results. The highly variable nature of the sequence between species not only allows for diagnosis, but also the identification of M. mycetomatis in soil samples. It has also been cited as the recommended method for isolation of M. mycetomatis. Additionally, molecular analysis led to the determination that M. mycetomatis did not share a common ancestor with M. grisi, and belonged in the Pseudariales class. Antifungal susceptibility. Understanding how a fungus might react to various antifungal agents in vitro can be beneficial when wanting to study or isolate particular organisms in culture. The Clinical and Laboratory Standards Institute (CLSI) M38A methods indicate that conadial suspensions are to be used when testing antifungal susceptibility of filamentous fungi. When following these methods for M. mycetomatis, alternative suspensions of hyphal fragments are required, as conadial forms are exceedingly rare. Table 1 summarizes the minimal inhibitory concentrations at 90%, MIC90, for various antifungal agents, with specific relation to M. mycetomatis. See also, Eumycetoma.